Hello student. In this session, we have to learn use of traveling microscope. Now let's see a traveling microscope picture. So this is a traveling microscope picture. So this is a microscope tube. Then this one is a screw which is used for fine adjustment, vertical adjustment. This is for fine horizontal adjustment. So this one is a main scale. Then one more main, this is a vertical main scale, and here this one is a horizontal main scale. Then after that, vernier scale. So it has a vernier scale also. This is a vernier scale. It is printed in the smaller part. So one more vernier scale is here, which is for horizontal reading. Now, after that, this is a clamp type screw. So this is a clamp type screw. By using that screw, we can rotate this microscope tube. Now, so this is a screw. By using that, you can move this microscope tube up and down. Then, so here, these are the leveling screws which are used to level the device. So this is here, a lens is given to note down these vernier division. Now let's see how to note down a measurement by using this device. You can go through this diagram, then you come to know that what is the use of which screw. Now after that, we have to note down one measurement. So you can see this is a diagram, and we have to note down the measurement means we have to see this part very carefully when we are taking vertical reading. And we have to see this part when we are taking horizontal reading. Now I consider that this micrometer tube is adjusted clearly for a particular, it is focused on a particular object, and when object is seen clearly, then we have to note down the measurement. We have to note down main scale, and we have to note down vernier division. Now listen, so you can see this one reading is shown here on this left hand side. So before going for the reading, actually we have to know its least count. Least count of this device, we can say that least count of device is given by formula smallest division on main scale. Smallest division on main scale divided by number of divisions. Number of divisions on vernier scale. Number of divisions on vernier scale. Smallest division on the main scale. This is the main scale. You can see here smaller part of the main scale is displayed here. So this is the main scale. And this part is what? Vernier scale. This is vernier scale. Okay. Now smallest division on main scale means value of the line which is coming after the 0. Listen here, after 0, this is a 0.1, this is 0.2, this is 0.3, this is 0.4, and this bigger one is 0.5. But here between 0 and 0.1, another one smaller line is there, and its value is 0.05. So smallest division on the main scale is 0.05, and the total number of divisions on vernier scale. So listen, this is a reference line on the vernier scale. After that, whatever the line is coming, these every line is called as division. Now, so first division, then second, then third, fourth, fifth division, 6, 7, 8, 9, 10. Up to 0.1 is written. Total 10 divisions are there. So up to 0.20, 0.2 is written, 20 divisions are there. Up to this 0.30 means 30 divisions are there. 0.4 is written somewhere, it is 40 divisions. As a reference, you can consider, and this is 50 divisions. Total number of divisions are 50. So 0.05 divided by 50 is 0.001 centimeter. This is a least count of this traveling microscope. Now we have to note down a measurement number 1. So whatever the reading, whatever this diagram is shown, by that diagram we can note down one measurement. We have to note down main scale reading. So note down, we have to note down main scale reading. Now main scale means what? This is the main scale, you know that which one is the main scale. But how to note down main scale? See the reference line. This reference line, with respect to the reference line, we have to note down main scale. Where the reference line touch on the main scale is the main scale reading. You can see here, it touch between this and this. Means here you can say this is 0.1, This is 0.2, 0.3. Okay, 0.2, 0.3, and this is 0.4. One line before 0.4. One line before 0.4 is what? 0.35. So main scale reading is 0.35. Listen, it is between 0.35 and 0.4. So it is 0.35, the lower one we have today. Now, after that, we have to see which line out of these 50 divisions is coinciding to the next line. The number of that line we have to write down, and it is our vernier division. Now, you can observe this very carefully, and I can find here this line. You can see this line is going straight to the next line. So number of that line we have to write down. Up to this point, point 3, 30 lines are there. Then here it is 35. After 35, 36 and 37. This one is 37 number line, which is going straight to the next line. So here 37 is the vernier division. Vernier scale reading we can write by multiplying this vernier division by least count. Or I can say this is A, this is B. Then vernier scale reading is B into least count. Least count is 0.001 multiplied by 37, 0.037 is the vernier scale reading. Now we have to add this main scale and vernier scale reading. Then we get a total reading here. You can see a formula is written. So here 0.387. After adding these two, we will get a total reading. So as we add this and this, we get a total reading 0.387, and this is the measurement by traveling microscope. I hope everyone understood how to note down this measurement. Only you must know how to note down main scale reading and vernier division. I hope everyone understood. Thank you for watching this video. If you really like this video, don't forget to press a like button as well as share it to see such kind of videos.